Thank you, Eduardo. And if there is any question in the audience, please let me know. I had a question. You quite rightfully insisted on the need for integration and basically standardization of the way we collect data. I guess one of the problems, at least from the academic point of view, that I see is every single instrument or every single company has its own way of collecting sometimes data or creating instruments that collect data with a particular format. Is there a way forward to actually set up a better uniform standard way of aggregating data that we could all use?
Yeah, that's a good question. So we've been kind of like as a company been looking at the number of standards for representing and sharing different data sets. Of course, like including early versions of the SFOL standard to encode designs, but more recently, we've been looking at others standards to exchange to represent experimental protocols and workflows. We've been looking at this of great interest of the industry. But yeah, you're right, like the industry, they are very, it's challenging, they are very sensitive to IP. So we're also kind of, but they still want to collaborate. We see, companies still want to collaborate, among each other or with academic partners. So we're looking at standards, for example, for sharing confidential data sets, for training AI algorithms, looking, for example, on some techniques coming from maybe some of you might know about fully homomorphic encryption schemes that can be used to encrypt and share experimental results, for example, to train machine learning algorithms in a collaborative manner while still protecting IP, from specific collaborators. So, yeah. But yeah, indeed, this is kind of like a challenge.
Thank you. In the interest of time, I suggest that we keep it here. Please do address your questions to Eduardo in the Q&A, and he can answer in writing. So I suggest now we move on to the next speaker, who is Hector Garcia Martin. He's the founder of Berkeley Labs Quantitative Metabolic Modeling Group, the Deputy Vice President for JBEI's Biofuels and Bioproducts Division, and also the Learn co-lead of the Agile Biofoundry. So Hector, the screen is yours.
Okay, great. Can you hear me?
Yes.
Okay, well, thank you for the invitation. Very happy to be here. And I'm going to talk today about how we are in both the Agile Biofoundry and JBEI and other projects in Berkeley Lab, combining machine learning and automation to try and make bioengineering predictable. And as you all know, there are high expectations for synthetic biology. This is a 2019 cover of Economist talking about the synthetic biology redesigning life. I don't know if we're doing that yet or close to that. But it's true that we in synthetic biology had an impact already in society. And Chris Ross had an interesting paper a couple of years ago on several commercially available products that are changing our world right now. It's 2 years old, and as the field moves quite quickly, it's a bit outdated. But we still have things like the Impossible Burger out there being sold at Burger Kings everywhere. There is also the Hyaline by Zymergen that has suffered from some setbacks, but still being used to produce these wearables and these polymers to produce transparent products and biomaterials. And the same thing, Kimbria, it's been used for a while, to reprogram T cells to combat leukemia and other diseases. So it is making an impact in synthetic biology. And to make it even more interesting, our capabilities are better than ever and growing better than ever. We have new tools that are really making the work of synthetic biology much easier, much more effective, and much more powerful, right? CRISPR, for example, allows us to do tens of thousands of edits and do genome-wide changes in cells. We also have DNA sequencing and synthesis capabilities that grow exponentially. This is the price for gene synthesis, and this is the price for oligo synthesis, and for over here, and here is for DNA sequencing. So we have the ability to synthesize more genes and sequence more DNA in an exponentially growing manner. And also, we're starting to see these high-throughput workflows for proteomics and metabolomics, which are maybe the second workhorse besides transcriptomics in terms of phenotyping cells. And this is something that I took part on, and we are starting to automate the proteomics workflows and able to do thousands of proteomes in a month, for example. So, and then there is also microfluidics that allows us really to do automated manipulation of cells and parts. And this is an example of how we can take different droplets with parts in there, mix them, move them as necessary, and mix them together to DNA assembly. Microfluidics is something that has a lot of promise. It's already being used in some commercial products, things like the Onyx or biolectors and so on, but it's not still in widespread usage, but it's certainly coming. So it seems like synthetic biology is really living an exponential growth, both in terms of publications, but also in terms of total funding raised by different companies, right? As you can see, up to 2020 until 2020, the investment in new companies has been growing exponentially. So it seems that we're living in very auspicious times, but not everything is perfect. There are still some problems, right? This transition from learn to design seems to be what remains the main bottleneck for high-throughput pipelines in synthetic biology. For design, as we have seen from Tesselogen and other people, there are things like Diva or Raven or the Tesselogen design tool module where you can choose your promoters, your genes, terminators, and so on, and you can create effective designs. Then they can be passed to build systems that can really benefit from this dropping cost of gene synthesis, and also we can use CRISPR to do genetic edits in a much more efficient way. So we can build the strains much more efficiently. In terms of test, we've seen that both the decrease in price of DNA sequencing for transcriptomics and the ability of high throughput workflows for mass spectrometry is really allowing us to really clear this bottleneck here, right? Oops. And then the issue where I see is most problematic is in learn. Typically, what we've used is our mechanistic models such as genome scale models and kinetic models and so on that would allow us to choose the next set of experiments based on our mechanistic understanding. The problem here is that no model is perfect and they fail, and that's expected and not bad at all. But the problem is how do we really take advantage in a systematic way of the fail predictions? So the ones that actually work, are great, but how do we take the new information that we get from the failures and then use it to modify the model to make it more effective? And our solution to this has been, well, to have someone think about it, typically a graduate student or a postdoc for 2 months to 6 months to maybe even longer to actually continue the DBTL cycle. And that has been the main bottleneck. Now, the advantage here of machine learning is that really machine learning is really the discipline that studies or creates algorithms that systematically improve with more data. So the use of machine learning here to really bridge this learn to design step really allows us to gather, to leverage all that extra data that we are creating with all these high throughput techniques in a much more efficient manner. And I'll explain to you. And that's why I'm very interested in machine learning and AI. That doesn't mean that it shouldn't include mechanistic insight. But the whole point of AI is that, the more data you have, the more effective you can become, and you can leverage that data effectively. Now, machine learning can be used in many places, right? If you look at the whole synthetic biology pipeline from looking at the societal need to choose which product to make, to DNA design, to creating the system, to then optimize it, to scale it up all the way to extraction, you could use ML in all of these places. And we have a review in metabolic engineering called Machine Learning for Metabolic Engineering, where we really go in detail about many of these applications, and you can have an introduction for how machine learning can be particularly used for metabolic engineering. I only have a limited amount of time now, so I will focus on a very specific example, which is going to be design of pathways. And this is based, what I will explain is these two papers that we published with Teselagen and the Center for Biosustainability in Denmark. The first one will be a new tool, a machine learning automated recommendation tool that is specifically designed for synthetic biology, for being able to use small amounts of data for recursive cycles and the need to quantify uncertainty. And the other one is an demonstration of using this ART, this automatic recommendation tool, to increase productivity of tryptophan. Okay, and this, as I said, was done in collaboration with the Novo Nordisk Foundation Center for Biosustainability and Michael Jensen's group and Jay Kissen's group there, and then Teselagen as well. And the reason that this project was really possible was that we have a new CRISPR method that permit this one-pot assembly of pathways, right? Or well, or in this case, we were able to do substitution of promoters for a variety of different genes, right? And what we did with ART is be able to, well, you can make a library in which each of these 6 promoters is added to your gene as desired. And what we did is use ART to choose which promoters to select for each of the genes that will lead to a maximum productivity. We did it only for 5 genes, but we think we could actually expand it to more than those 5, right? And the way that the reason that we did only for 5 is that because the combinatorial explosion really becomes really big. And this was a pilot project to show that machine learning can really lead metabolic engineering. The 5 genes that we chose were chosen for a genome scale model that allowed us to pinpoint which of those reactions were most important for creating the final product, which is this tryptophan. We're going all the way from glucose to the glycolysis and pentose pathway, all the way into tryptophan. And the procedure here was to change the promoter for each of these genes. And we had a selection of 6 promoters for each of them. And it wasn't entirely clear which is the best combination for these promoters to get the maximum productivity here. If we did all the 6 promoters, we'll get around 8,000 combinations, which is starting to be a bit challenging in terms of phenotyping. So what we did in this case is we used the automatic recommendation tool, or ART, to train on a variety of on data on instances that have a certain given certain promoters. So promoter 1 for gene 1, promoter 7 for gene 2, promoter 15 for gene 3, and then the associated response, which is the tryptophan productivity. We only did 264 combinations out of the 8,000, and that's only 3.4%. But with this training data, we were able to then, we were able to measure the tryptophan productivity with a biosensor, because measuring the tryptophan through an LCMS would be challenging. So we were able to, we were happy to have a biosensor that would enable this high throughput measurement of the phenotype. And then with this data, we were able to train a predictive model that was probabilistic. So I'll tell you this promoter combination over here has a 10% chance of producing 10 milligrams per liter, or a 20% chance of producing 50 milligrams per liter, and so on. And then based on this predictive model, train only 3% of the total possible amount of promoter combinations, we're able to make recommendations of promoter for the next cycle. Now, as usual, when working with bioengineering and biology, there is going to be a lot of data attrition. Initially, we wanted to do 96-fold plates, around 600 strains that will give us around 1,700 instances to train our algorithms. But of course, things went wrong at many places. In some cases, when we sequence the strain, we didn't get a complete genotype information. We couldn't do the full sequencing. There were also errors in the assembly, in putting all these promoters together. Some of them didn't pass a minimum growth threshold, so we can get data on them. sometimes we couldn't get the complementation plasmid, and sometimes we didn't get a single population per clone. So that lowered our 1700 instances to a final of 792, which are 264 combinations in triplicate, which are around 3% of all the possible ones. Now, it's important when you're doing machine learning and synthetic biology to choose what you're trying to optimize, because that's what the algorithm will really give you in the end. In this case, we were trying to improve the productivity, the tryptophan productivity. We chose 2 points that were chosen in such a way that we wouldn't have a lot of lag effects here, but also it wouldn't be affected by the oxygenation problems later on. So we choose a couple of points here. We measure the change in the GFP that is proportional to the tryptophan divided by the time, and that gives us where we're trying to optimize for all of them. When you look at the data, this is what you get for the productivity. This is for first gene, second gene, third gene, fourth gene, fifth gene. And these are each of the promoters. And as you can see, there is no real trend, right? If you choose the first promoter, the productivity is all over the place. The exception here is in gene 3, where you can see that choosing promoter 16 or 17 really give you an increase in production. But what is important to realize here is that by looking at this data, it's really hard to figure out that the combination of these, this promoter with that promoter, with this promoter, and that promoter, and that promoter will give you a higher productivity, right? You need an algorithm to figure out the nonlinearities and make final recommendations. And that's why, and that will become even more complicated if instead of 5 genes, we had like 10 or 15, right? And the machine learning or art in particular, and the machine learning methods are really effective in taking that data and deconvolute it. Now, the other thing that is really important is to get high quality data. And in this case, what we mean by that is that there is significant reproducibility, right? In this case, this is the replicate 2 versus replicate 1. In the perfect replicates that will all be on the line, you'll see that a few of them, like this one, are really off. It's important to eliminate all of that. We took out 5 strains because we had these irreplicable data, and that can really throw off the machine learning techniques, right? Unlike other models that use more mechanistic information, the machine learning or data-driven models really rely just on the data. If there is a bias in the data or no replicability, that can really lead you in the wrong direction very easily. And then based on that training information, Art spit out all these 15 recommendations. And the recommendations are the promoter that is expected for, or that is recommended for each of the genes. You see this, for example, this 16 promoter that we saw over here really give you high production is the one that is recommended for all the third gene for a reason. And besides the recommended promoters, where we get to is productivity, expected productivity, predicted productivity, and the whole probability distribution for that. This is the maximum obtained so far. So if you integrate this tail over here, it'll give you the probability that you get an improved productivity with respect to the training data. So how did it work? Well, it worked pretty well. We were able to, well, if this is the predicted productivity and this is the observed productivity through the fluorescence proxies to the GFP, this is the base strain where we started. This is the whole strain library, and this is the best strain in the library with the 260 strains that we use for training the algorithm. And these over here, the recommendations by Art. And you see that there are recommendations to increase the productivity of the best strain in the library by 17%. And the final recommendation really increases the productivity by 105%. So, and this is with the GFP. Of course, all metabolic engineers wanted us like, well, we wanted to see not just the proxy, but the LCMS. So for a few of them, we actually checked the LCMS measurement of tryptophan. And we were able to see that for the base strain, actually none of the library were able to increase the base strain, because particularly for the base strain, the linearity between the proxy and the tryptophan did not hold very well. But what we did see is that the recommendation by Art really increased the productivity by around 43% for the best in library over here. Now, this was only one DBTL cycle. I think most machine learning methods and art, we believe, will get better and better information and better predictive capabilities with more DBTL cycles, and that will help us increase this more systematically. However, this was enough for the grad student involved to get a paper, so he published it in Nature Communications and got on with his life, which is understandable, but left us a little bit desiring to do this with more DBTL cycles. So in summary, art here, the automated recommendation tool, bridges these learn and design phases of the DBTL cycle. So if we, this is the DBTL cycle we have at JBEI and ABF, where we use DIVA to design strains, and we build them in a semi-automated way. We culture them and phenotype them through proteomics, metabolomics, or other types of omics, put that into our data deposit. And we draw the data, we take the data out, put it into art, and art gives you the recommendation. So it's this part here from test all the way to design that art does really, really well. And there are many algorithms out there, machine learning, that are able to predict outcomes. But I think the extra step in art that makes it interesting is that it also uses those predictions to recommend the next set of steps. And we've proven that it gives a significant results. And we've done this in a variety of other systems for media optimization and others, but those are not quite ready for publication. For those of you who are interested in how it works, how art works in the inside, I'll just tell you that it's an ensemble model where we have several machine learning models, mainly from scikit-learn. Each of them give you an output, and then we do a Bayesian ensemble model where we linearly add all these results for each of the models, and then we decide these weights for each of the models based on how productive they are. We use Bayesian inference, and if let's say model 1 is particularly effective, it has a large weight, and if it doesn't, it does not. And in the end, that gives us because these weights are the probability distributions, then we get a final predictive distribution that gives us the probability of getting a given productivity. Now, these involve all the scikit-learns, random forest, all the scikit-learn models like Gaussian processors, random forest, network gradient boosting, and so on. And for that reason, the final model is able to outperform any of the single models, including the Gaussian processor, which is the one that is typically used in recommending next steps, right? In this kind of affairs. In terms of the optimization, which is an important part of art, what we do is once we have the predictive model that we talked about, we do a global optimization in which we do a Monte Carlo sampling through all of the possible promoters. And we choose the ones that have the highest predictive production, but also the highest model variance. That is the place where the model is most likely to fail, and parts of the phase space that we like to explore more to make the model more effective. And these recommendations here are the ones that you see finally in the final recommendations. Now, if you're interested in art, there is a whole artworks website, art.lbl.gov. There you will find some information on art, how it works. You'll find tutorials for simulated data and real data, and even some papers that show you how to use it in combination with eyes, EDD, and a variety of other tools. If you are not so much into programming, there's also art front end where you can upload your data and they'll get your recommendations. And now that I've shown you, well, how ML can really impact synthetic biology, and I gave you an example with art, I really like to go into something a bit more general, which is, machine learning or AI can really make a difference in how we bioengineer cells and how we do synthetic biology. But it really needs a significant investment in infrastructure. It's not just a magic wand. What we typically hear about is, this part here, the deep learning, machine learning, and that's what you see on the media and what people talk about. But in order to make this possible, there's a lot of layers that have to be created in order for this to be effective. Before you go into deep learning, it's very useful to use some very simple ML algorithms like logistic regressions and so on, do some feature selection, basic testing, and so on. I spent some time in cleaning outlier detection. Also creating data standards, ontologies, and databases, because you're going to generate a large amount of data. So you want that to be in a system, let's say in a standardized way, so you don't have to deal with like 500 Excel sheets that collect data in a different way. For that purpose, we created something called the Experiment Data Depot, EDD, that allows you to really store your data in a standardized way. And there is a paper here. You can see more of that here. You can go to public-edd.jba.org and you can get your own account there, and you can start putting data there. Another important layer that comes even before the data structuring is the collection of data. And as I said, we need high quality, abundant data for high quality data for machine learning. And for that, the best way to avoid the biases that humans usually introduce is to try to automate as much as possible and really do some careful experimental design. In that sense, we've been working on automation and high-throughput analytics for a while. And my personal opinion is that as this trend continues, the future really involves what is called self-driving laboratories. That really makes the automated robotic platforms that do the experiments with the AI and machine learning algorithms that take that data, those measurements, and decide where are the next set of experiments to do, right, through a series of control software. This has already been done, by people, this privacy group and other people in Canada for materials, for thin film materials. In this case, they had a robotic platform that did this cycle, which they were trying to look for different materials. They prepared a precursor solution with different stock solutions. They cast it into a thin film. They annealed it with hot air, and then they record the image of the film. They phenotype the materials, as we would say. They record the optical spectra, and then the field conductance that they're trying to use to calculate the whole mobility, which is what they're trying to optimize, and that goes into parameter into a K-mouse, which is their algorithm that determines where is going to be the next experiment in terms of which stock solutions to use and how to annihilate and so on. What we are working on, which is similar to that, or it goes in that direction, is using microfluidics to genetically edit cells, and for that we're using a CRISPR system where we have a variety of different guide RNAs and templates that are able to edit in a different part of the cell genome and in a very specific way. And from that, we're able to measure where's the production through fluorescence. So they will have an improved production with respect to wild type or decreased or so on. And the idea is to use this information to then do the next experiment and have these cells go to the next gene editing. So far, we've done a proof of principle of this system, and we're able to do 5 genetic edits that have a measurable or learnable effects in production. In this case, we had an indigo-iodine synthesis that was taking glutamate into glutamine and then created this indigo-iodine dye that is a blue pigment over here. And we took the promoter and we made modifications. So in this case, this is the original strain with this amount of production. If you make this modification in the T7 promoter for the indigo-iodine synthesis for the NRPS, then you have a decrease. But if you do changes in the promoter for the glutamine synthesis, then you see that some of them don't make a difference, some of them seem to be a small increase, and finally the last one is able to increase production. Now, we think this is learnable. We are doing now hundreds of edits. We hope to be doing thousands soon, and we think that we can learn this sequence to function map and then recommend new designs to systematically increase production. This is already finally published in Microsystems and Nanoengineering, and you can see the first results of that. And we are hoping we can do soon thousands and do a quite quick DBTL cycles on the order of 2 to 3 weeks. And that is that's mainly what I wanted to talk about. Just wanted to say that really machine learning and automation can expedite synthetic biology solutions. Synthetic biology can produce a lot of interesting things, and we're still trying to figure out where those things are and how they can help society. But the problem is that synthetic biology really, it's really hard to predict how or design things to a specification, right? We basically try a variety of things and see what happens. So I think machine learning can provide the predictive model to do this inverse design. So if you want a pathway of a strain that creates tryptophan with this amount of productivity, we should be able to use machine learning with data to predict how to do that. And the only way to make machine learning predictable is by feeding it enough high quality data through automation. And that's, I think, the future of synthetic biology or bioengineering, in my opinion, to use automation to produce the data needed for machine learning and mechanistic models to be predictive, so synthetic biology can produce the products that we need. And that's it. These are some of the products that we've seen, and there's going to be many more. Some of them will fail, some of them will not. But it's imperative that we're able to produce more and more effectively, and they don't take hundreds of millions of dollars and 10 to 15 years to make. But we should be able to enable companies to make new products with a few $1 million, and in a few months. And that's, I think, where we should go. We need for that predictive power, which I think can be provided by a combination of machine learning and automation. And finally, self-driving labs in that sense, in combining machine learning and automation, really provide completely new capabilities. And that's all I really wanted to say. Final thing is, well, this is my group. We've been working on this for a while, but we wouldn't be able to do this if we weren't in the national lab, in the Berkeley National Lab, and we do teamwork with a multidisciplinary team that involves people from synthetic biology and informatics to microfluidics, host engineering, metabolic engineering, automation, and a variety of other capabilities. So it really takes a team, and it really takes a multidisciplinary team to make this happen. That's it. I'm happy to take any questions you may have.